starting that now. We're recording these and we're putting them up on YouTube, so if you are worried about your face showing up online, you can turn your camera off. And if you do have any issues, please just drop me a message. Down on the right-hand side in the Zoom chat, you'll see people. You can feel free to discuss amongst yourselves any issues that come up during the chat today. If you have any questions that you want to pose to the crowd, please feel free to use that and socialize. Be relax, and this is meant to be relatively lighthearted, so please do make use of that. If you haven't used Zoom before, when you open up the participants list, you'll see on the bottom right-hand side, you'll see a raise hand option. And when we do get to questions, if you could use that to make yourself known to me, and if you have your camera on, you can also wave your arms frantically, and should be able to see you. Of course, as well, you can, as I said before, if you do have questions that we don't get around to, you can use the Zoom chat option there. And if you have any other technical issues, please just try and drop me a message. I know we have had some issues with people connecting in the past, so just let me know. All right, and with that, I think I'll hand it over to David to start us off with the first paper. David, are you there?
Yes, hello everyone. And we start with a very neat paper on coordinated motion in seabirds at sea. Hoping that Camille, Assali, the lead author, is online with us. She's been leading that study performed by the Marbec lab in Montpellier. And we picked it up because we thought it was really relevant to a long-standing discussion about the use of public information in seabirds. So of course there are quite a few papers about public information, but actually more speculation than data. And those papers mainly focus on processes on land at the colony level with habitat selection. So there are historical papers on this by Danchin, Bouligny, and colleagues. But far fewer papers about what happens at sea in terms of foraging behavior and the use of public information. There are direct observations of coordination at single and multi-species feeding aggregations. For instance, you've all seen directly or through footages groups of shearwaters or shags coordinating their dives to push small pelagic fish to the surface. But it was really unknown of what happens at larger spatial scales between seabirds. So what Camille and her colleagues did, they used images captured by radars, which were on fishing vessels off the Gulf of Guinea in West Africa. And of course, another question would be, what French fishing vessels are doing taking fish off West Africa, but I won't get into that debate today. But the technical fact is that these fishing vessels, they use a bird dedicated radar to see the birds at sea and to deduce the presence of surface feeding tuna so that they can target this tuna. So Camille and her group worked on these images, and I was really impressed reading the paper by the amount of post-processing which was necessary to convert radar data into something they could use to track and identify the birds. But at the end of the day, what the authors showed was that there were very distinct coordinated flights of seabird groups. And those took the form of seabird fronts, so rake patterns of up to 11 birds at up to about 4 kilometer width. And this front of birds was moving for about 4 kilometers at about 30 kilometers per hour. And the authors think that the probable species were sooty terns, brown or black noddies. And these birds, they were not close together. They were about 500 meters from each other, which means that they could see each other, of course, because seabirds at sea can detect the presence of conspecifics as far as 10 kilometers. But that depends a lot on at sea conditions. And this whole study implies that in these birds of West Africa, there was a kind of collective game with little room for cheating in prey searching, because as soon as one bird would stop to feed, the others would converge. And it was thought by the authors that maybe they would even communicate vocally when finding prey, as suggested, for instance, in gannets. So I think this study is very innovative. There aren't that many radar studies at sea to track seabirds. And of course, there are still many unknowns, but the rake patterns, as shown in the papers, are pretty impressive and pretty convincing. But my first question to Camille would be to try and guess how often these patterns actually occur. Do you think, Camille, that this is happening really daily within your study zone, or does it occur with specific at sea conditions? I know, for instance, that you used radar pictures when the weather was really good at sea.
Yes, because, so hi everybody, thank you very much for this presentation. Can you hear me well? It's okay?
Yep.
You're good.
Okay. Okay, cool. So yeah, in fact, I saw raking patterns in almost every day of a year-long data collection, and kind of off the coast, and except of course days with bad weather, because the radar data cannot be exploitable with rain. But yes, we were astounded by the number of this formation. We could see maybe, I don't have a proper rate, but several rakes per hour at some times, but all year long. And in this study, we only collected the rake patterns that were very visible, because we visually collected them on the radar images. So we chose the best ones. But maybe I can share, I will try something, share my screen to show you maybe the radar data.
Yeah, you should be able to.
Yeah, because I think it can be inspiring to see these collective patterns. Okay, I can try. Check.
Yeah, and that's good, because probably the PDF was not available for everyone.
Yeah.
Yes, so here we can, okay, I try something. Can you see the GIF? No. Yeah, can you see the radar images now?
Yes, yep,
Yep.
we can see it fine.
Okay, so maybe it will be easier as we see the kind of data I treated. So you have a radar disk, and in the center of this disk, you have the boat with a saturated zone of 1 to 3 kilometers around it. The range of the radar disk is here 8 nautical miles, so approximately 16 kilometers. And what you can see that are the orange dots all moving around the boat are groups of seabirds, and some boats, at sometimes the most elongated ones, and the most large, yeah, the largest one here in the upper part of the image is a boat. So it's
and I can't remember whether you wrote this in the paper, but do people actually see these patterns from the fishing vessels sometimes?
No, the rake patterns, in fact, they don't. Sorry, I may not understand your question. You can see it on the radar image.
No, I mean whether from directly from the ship through direct observations, do people sometimes see these rakes of the groups of birds progressing something like 500 meters from each other?
In fact, they are on the boat. They are mostly fishermen, so they target especially aggregation of birds, but they don't very take attention to the collective flights like rakes. But they might be able, because they have very powerful binoculars on board. So if somebody at the radar could tell them to check what species is flying that direction, they could check, I think, with the binoculars. but all people in that boat are fishing, so they don't care. But yeah, they only focus on the.
And the other thing I wanted to ask you, so you say that the rate moved at about 30 kilometers per hour. And I looked, in that classic paper by Spear and Ainley on the flight speeds of birds. And for the terns, I found something which was half of that value. So I was wondering whether you had direct observations under these conditions of the flight speeds of terns and noddies, which would confirm, that they moved with that speed, especially if the weather was really good. That seemed quite a lot.
Yeah, I understand. We have no complementary data. We had only radar data at that time, with no observer on board. So very frustrating also because we don't know the species and etc. And the number of birds within an echo can be 1 to 10. Yeah, but yeah, we had no direct observation to confirm that. And yeah, I cannot tell you more, but
okay.
Yeah, sorry, maybe a source of what can be. No, I don't have. I don't know how to explain. Yeah, but when you have an echo, sorry, you have a centroid, and our trajectories are based on that centroid. So when it moves, yeah, the speed can be a little bit false and altered by this centroid that can move within one echo. But yeah, approximately the speed was 30 kilometers per hour. And since we don't know which species was in it, yeah, it's still.
Another study which did the GPS tracking of sooty terns, and they had the flight speeds which were relatively low, but I think they were taking only one position per 20 minutes. So it was hard to get speed from that. And the last thing I was wondering about is for these radars, is there a lower limit to the size of seabirds they can detect? Because these terns and noddies, they're pretty small.
So yeah, the radar technology makes that if your bird is within the beam, it can be well detected close to the boat, but with that beam that enlarge with the distance, if you have two birds in that beam, they are merged in one echo. So for terns, individual terns, we could see them if the radar was really, the range of the radar observation was very narrow, like maybe 1 or 4 kilometers. But as here we have 16 kilometers of range, yeah, that's true that we can miss individual small birds, but we have big ones or groups of small, but it's a general idea.
Okay, yeah, I get it. Well, congratulations on the very neat study and especially the really advanced analysis. And with this, I'll hand over to Grant in case he has questions.
Well, yeah, you took the one question that I had, so I'm gonna have to come up with another one on the fly. But I guess I'm curious as to if you were able to detect maybe more fine-scale behavior. Could you tell when birds were foraging, for example? Could you tell when they were just loafing around? I assume you can't really see them on the water; they have to be flying, correct?
Yeah, exactly. Because the ones that are on the water are filtered out by the radar box, because you won't see anything with the waves, all that. So what is on the water cannot be seen with that kind of radar. And now to have more precise behavior, you have to really reduce your observation range. So now we can only see the range of collective behavior, of group behaviors. And yeah, even we were not on board to check if they were foraging or just flying, or yeah, but they are more than 100 kilometers from the coast, so they are probably foraging. And we see very few reasons for them to be only transiting, or yeah, they were, I don't know.
Yeah. And so could you potentially see, I know you had mentioned a little bit sort of about the size of the birds, but would it be really obvious to say, tell a difference between, say, a gannet and shearwater?
Me, I don't have the experience with the radar images, but I know that fishermen that are spending all their time in front of this radar can make a difference between some group of species. And I've observed some specific behaviors for groups of seabirds associating with dolphins comparatively to tunas. So they can detect that, but I don't have now the experience and the eye to detect it myself. And then they also detect the behaviors of frigate birds that sometimes act as catalysts. And so they see some big bird waiting for something, and then they see other birds just joining in and making a big aggregation. So they're also targeting that. But I really miss the fisherman experience to make this study more complete, because they really are the ones that have the true observation experience.
Is there any way of say tracking individuals across this? So I mean, obviously the radar is sweeping, so you do have a break in the images, but is there any way of, say, pinning a centroid to each of these birds and then following it through the radar track?
So is your question that you can miss sometimes some birds and then track them again?
Yeah, so like if you obviously you see a sweep, you see a group of birds, can you identify each of those, like put an ID value to each of those, and then track them across the various radar sweeps? Is that possible?
Partly, in fact, one of the procedure of radar analysis is to reconstruct the tracks of vehicles. So we have some algorithm that can deal with the fact that sometimes the birds disappear and reappear and disappear, yet we cannot target them when they merge within a group and then split again. So here we are totally lost the individual track. But yes, we can deal with some momentaneous disappearing of the echoes.
Yeah.
And that's all.
Cool. All right, we'll open it up to the audience here. We've already got a question from Abdullah Hamza. I'll unmute you, my friend. And there you go. Go for it.
Yeah, hi. Good evening. It's midnight in Malaysia. So
you're one of the brave few to stay awake.
I missed the last two. So because I always confused about doing my reminders, but this time it worked. So just to ask, you worked on a boat and you used the radar from the boat. So are the fishermen that you worked with using these aggregations of seabirds to move to the next fishing spot?
Yes, exactly. In fact, when they observe one aggregation, they suppose that there is an aggregation of tuna that is just above the surface, and so that tuna are catchable. And so they really target some behaviors like these aggregation to move and choose their probable catching maneuver.
Mm. Right, and do you have any idea what species they use it as an indicator for that fishing spot? I'm sorry, it's out of the topic, but it's quite interesting as well.
Yeah, I would have this example of the frigate birds that are catalysts for some aggregations, but no, they really treat the information of the behaviors on the radar image, but not target some species especially. They really look at this large scale of some 16 to 30 kilometers, and when they spot something interesting in the radar image, then they really direct on it, and then they check with the binoculars if a tuna is on the surface. But they don't really mind about the seabird species, from what I know, but I cannot tell more precisely about that.
Cool.
Sorry for my English.
No, you're doing great. You speak better English than I do. Hey, do we have any more questions from the audience? We do. We've got Sam Ermy. I'm going to unmute you, my friend. Here we go. You're up.
Hi. Yeah, first I want to say I really enjoyed the paper. I was wondering, I've done some work with seabirds and radar before, and what were the sea conditions, the waves like most of the time? Kind of in my work, one of the biggest obstacles was getting reflections from the sea surface that interfered with getting echoes from birds, and I was wondering how much of an issue that was on these fishing vessels.
Okay, I'm not sure to have the question, but was your question how the noise from waves was filtered out by the...
Yeah, how bad was it?
How bad was it? I don't know how, but with the radar box on board, they could filter the noise from waves. And the most bad weather situation we had was very large rainfalls. And so now you can really see that the fisherman is just looking to something else and letting the radar doing its stuff and waiting for some better conditions. So yeah, we had a lot of images very saturated in signal, but rain was the most problematic thing.
Cool. Thanks, Sam. All right. Do we have any more questions from the crowd?
And in the meantime, maybe a tiny one, Camille. So you stated that the actual dots you see with the rakes, that those could be individual birds, but also several birds close together. Is that right?
Yeah, you're right.
Yes. Okay. So it could be actually both, huh?
Yeah, really. Yeah. So in raking patterns, it can be the same or several species together.
Because especially when you watch sooty terns over a tropical sea, very often they are in these small groups, maybe 2 or 3, and quite close together. So I was wondering whether you could have subgroups which may appear as dots and rakes on your picture.
Yeah, that's the point. Yeah, and you can really see maybe in the animation I made that some echoes sometimes split, and you can see that in fact it was made of 5 or 10 individuals.
Okay.
Cool. Any more questions? All right, I don't see any. Nobody's waving frantically at me or throwing anything at their screens, so I think I'll just go ahead and thank you very much for being with us, Camille.
Thank you.
And
thanks a lot.
We'll move on to our next paper. Now we're moving on to penguins. As you can see, I've actually brought a Gentoo penguin with me for this one. All right, so selection pressures can vary drastically between species. It can be driven by a number of factors, both abiotic and biotic in nature. Interestingly, during this global pandemic, we're going to discuss a paper that studied how pathogens can act as a force of evolutionary selection. Now, I know this isn't quite the same as coronavirus because we're unsure how, say, COVID-19 may lead to large evolutionary changes in humans. However, for somewhat isolated populations of birds, say Gentoo penguins, pathogens could act as a major force of selection, and that can be detected through genetic analysis. And this is what this study led by Hila, Levy, and Oxford aimed to investigate. The authors retrieved 155 genetic samples from a very strong geographic representation of their breeding distribution, about 14 sites in total, spreading across their entire range, which is quite impressive. Genotypes for 3 toll-like receptors were sequenced to infer the respective haplotypes for each individual. Now, toll-like receptors, for those of us who aren't genetics people like myself, are pathogen detectors. They do things like initiate inflammation to prevent infection, further infection. And of course, Hila, feel free to correct me on any of this when we get around to you in just a few moments, because like I said, I'm not a genetics person or a disease person, so my interpretation of your paper will be interesting. So these 3 particular TLRs that were chosen because they represent viral and bacterial TLRs that are present in all vertebrates. The authors also did some mitochondrial DNA analysis to look at population divergence and phylogeography. Now, the mitochondrial DNA analysis seemed to identify 4 regional clades. We've got one sort of southern clade south of the polar front, including South Georgia. Another clade in South America and the Falklands, Malvinas. a third in the Kerguelen Plateau, and a fourth consisting of birds from Marion and Crozet Islands. Overall haplotype diversity was different between TLRs 4 and 5, with both of those showing high levels of heterogeneity in the Indian Ocean, South American Falklands clade. Interestingly, the other one, TLR7, which is associated with detecting viruses, seemed to be relatively homogeneous across all populations. When they went further to look at, say, how these codons expressed selection pressures, what they found was TLRs 4 and 5, which were the most heterogeneous, were found to exert positive selection, suggesting potentially that bacterial pathogens may be playing a more important role here than the viral pathogens. Interestingly enough, the suggestion in this paper is that sort of densely populated Gentoo penguin colonies with low haplotype diversity of the TLR 4 and 5 and 7 codons may be more susceptible to disease. And so the sort of conclusion here is that extra care must be taken by, say, humans or visitors at vulnerable colonies to ensure there's no transmission of bacteria or viruses. Now, Hila, please feel free to elaborate on my interpretation there. As like I said, this is stretching well outside of my comfort area.
No, it's wonderful to see somebody read something that you put years into. So thank you for at least making a really strong effort there. I also really want to thank all the co-authors that contributed, because the only reason that this was possible was a huge collaboration from a lot of different samplers and research groups around the world that contributed either the genetics data itself or the samples for us to process. So thank you and some of them are joining us today, so I really appreciate that help in getting us to such a large study. One of the biggest challenges with Gentoo penguins is that they're the widest ranging penguins. They have the broadest range of any penguin species, and they span about 20 degrees of latitude. So we've always had questions about what kind of pressures are leading to them to evolve in different ways, and whether there is any differences geographically that are found. And we knew from previous studies, there have been a few studies on mitochondrial DNA or microsatellites, we knew that there was some differentiation, which you might expect because they don't migrate very far. They're philopatric and they stay within their natal colonies throughout the year. And so we wanted to look a little bit deeper at a functional gene. So this study came about through a collaboration, kind of expanding my population genetics interests with one of my supervisors, Adrian Smith, who is an immunologist, an avian immunologist. So a lot of work on chickens. And so don't worry, I'm coming from the same, I didn't know a lot about immunology, some genetics, but not immunology, and kind of coming into this to learn about what something in the avian immune system, how that might be affected by such a broad distribution. And so we didn't expect any of the necessarily to find something this interesting, I think, when we embarked on this project. So it was great to have such a huge input of samples and sites to evaluate. We knew that there was a likelihood that things like the polar front or the separation of space and across breeding cycles might have an impact. But when we actually did the study, we found these differential patterns because we had these 3 different toll-like receptors that, like you said, help bind to a pathogen and recognize it, and based on the pattern that a pathogen might present. So TLR4 and 5 recognize different bacterial pathogen components, like lipopolysaccharides, so one of the membrane components of bacteria, or like flagellin in the case of TLR5, so for flagellated bacteria, and TLR7 does viruses. And so we ended up finding very different patterns in those, like you mentioned, where TLR7 was not as differentiated. So we thought that weak signal might be what we would find overall. But in TLR4 and 5, the differences were really strong between the colonies and then across the polar front. And what was even more surprising, because it was, to our knowledge, one of the first attempts to try to do this, and this is my co-first author, Stephen Fidemann's work that presented, that actually carried these experiments out, was expressing these polymorphisms that we found, which we knew through modeling were quite likely to show some kind of selective indication and signal. But when we actually expressed them in cells, they responded very differently, significantly differently to flagellin, which gave us that indication that this has a real significance and may contribute to over the long term how these different populations are responding to bacterial pressures that are present in the environment, whereas maybe the differentiation across viruses is more consistent across the different seabird habitats. So I think there were a few things that we learned that we came out of this, and one of those is that we need to know a lot more about the pathogens that exist in seabird habitats and how these are shared across sympatric species. Gentoo penguins obviously don't live in isolation, so they often share their colonies with Adelie penguins or chinstrap penguins, or with flying and migratory seabirds and skuas. So whether these pathogens are transported by flying or migratory seabirds. And we also now know that there's functional genetic markers that can help us understand these interactions with biotic factors in the environment in phenotypic form. And so immune markers are just one element that can help us understand host pathogen interactions, for example. And we hope that we've also provided a tool to help others carry out functional experiments to express polymorphisms and then refine that approach from theoretical to having something more practical in significance, but still certainly a lot to learn and to know about what these environments hold for seabirds.
Yeah, it's totally fascinating stuff. What I really found interesting was this, in your mitochondrial DNA work, where you've identified these 4 different clades, and I know that it's sort of been suggested before that there could potentially be some speciation occurring, at least at a subspecies level. And do you think that what you found has sort of reinforced that?
I think we definitely hope that it provides more evidence of that, because now it's not just neutral markers that are being differentiated, which could be more of a signal of genetic drift, you would think, over time. But this is something that shows that no, the environment and the interactions that these penguins are having with their environments is leading to functional differences, which should support, with greater research, but this should lend some support to the fact that we should consider recategorizing the species complex. For those of you that are not familiar, it's a little bit of a debate in the Gentoo penguin research community, because right now there's only 2 classified subspecies. And I'll try to maybe share, maybe I can share my screen and show what one of these differences look like. I hadn't really thought to do that, but yeah, north and south of the polar front, there's a northern subspecies and a southern subspecies. But as we can see, Kerguelen and then the northern Indian Ocean also have quite a lot of differentiation, and they retain a lot of diversity, not just from neutral markers, but from functional markers, as we found in this study. So they might be the original potential source population evolutionarily for where Gentoos got their huge distribution worldwide. So I think this lends some support to reclassifying the complex, and hopefully bring some more attention to the differences that exist.
Yeah. That's an interesting point, actually, and it kind of brings me to my next question, which is about the polar front and this differentiation you get. In the TLR4 and 5 codons, it seemed like south of the polar front there was a lot of homogeneity versus north of the polar front. And do you think that may be related to perhaps just greater diversity of pathogens in warmer climates?
So we're trying to investigate that further. That's actually the majority of what my PhD thesis is about, is looking at pathogens overall, enteric pathogens, viruses. There's some really good new papers out there that have shown increases in RNA virus diversity just in the last week or so. I've certainly found, not published yet, but I'm working on it, a lot of pathogens that you wouldn't expect to find in Antarctica we're starting to see turn up more in microbial analysis through bacteria. So I think there's just a lot of hidden diversity that's present in Antarctica. It's hard to say right now whether that's strictly different between the warmer areas and the colder areas, but there's certainly a lot more underlying diversity in the Antarctic region. And so what we found in this particular result was that the southern, less diverse areas were clearly refined to have some sort of polymorphism that reacts in a stronger way to flagellin. So it's difficult to say what physiologically that might mean, because it might be that the immune system is being activated more strongly. And so just like with coronavirus, right, your immune system can do more harm than good. So we can't really infer whether what the strength of that differentiation might mean and what the actual antigen is that's causing that. Aside from flagellin, we don't know what the source might have been for that such a strong pressure to evolve in southern species, in southern subspecies, in southern colonies.
Mm-hmm. Yeah. Oh, very cool. And I had one more question for you. Actually, it's going to your analysis of the population size versus haplotype diversity. I was interested in wondering whether or not you also tested, say, population trend versus haplotype diversity, in under the assumption that maybe colonies that are growing more may have more diversity.
So yes, we did. We did look at that, and that's something I can try to pull up. But basically, we didn't find a correlation necessarily between, because we did have questions about how sample size might have a difference, and how that might differ relative to the haplotype diversity that we found. But it didn't really match up because, and let me try to share my screen and pull up the figure. How do I do that? Is it? I think I did that. Okay, I think I found it now.
Okay.
All right. So if I can.
Here we go. Yeah, we can see it there now.
Okay, let me try to find the right one. So if I full screen that, is that not what I was looking for? Sorry, everybody.
That's all right. We get to look at gorgeous scientific graphics.
Okay, so I'll try to pull this one up if you can see here. So one of the colonies that we had was from Martillo. So thanks to Andrea Rayare and her team for providing us with those samples through Juliana Viana and her team that did the analysis that actually sequenced those. But Martillo Island is just off Tierra del Fuego in South America, and it's shown by MT here off of South America. So that MT colony had only 8 individuals. Sorry, the whole colony itself is a population of 8 breeding pairs.
Oh, that's the entire okay.
The population varies from 8 to 12 individuals.
Yeah, okay.
Over time. And we actually sampled about 5 or 6, I don't have the exact number right now, but between 5 or 6 in the paper were analyzed. So it's a very good representation of that population, which is not that big. But if you look here on B on the left side, Martillo had a massive amount of haplotype diversity for TLR5. So there wasn't always there wasn't any clear correlation sometimes between what we would expect to find in very large populations where, for example, the Falklands and South Georgia have very large populations of Gentoo penguins, and yet South Georgia, designated here by Bird Island in the graphics, BI, which had a larger sample size. BI didn't necessarily, Bird Island sometimes had absolutely no haplotype differentiation whatsoever in some of these colonies. So there wasn't necessarily a clear correlation between the population size present and the result that we found. So it's hard to say that trends, what the trend would be on growing versus shrinking. We know that some colonies, for example, at Marion are in trouble. We knew that there have been declines in the Indian Ocean for Gentoo penguins. From a conservation standpoint, for example, even though Gentoos are designated as least concern right now overall, there's still very big differences in how each particular part of the population is faring. Probably another argument for why there should be different management categories, potentially more subspecies considered for those. But in the Indian Ocean, on Kerguelen Island, which is designated by both the MO and the COU here in the Indian Ocean, those were very high diversity, even though they have experienced significant declines in the last 30 or so years.
Yeah, yeah. Oh, very cool. All right, David, I'll hand it over to you for some questions.
Yeah, maybe that relates to what you just showed, but as you know, even within Kerguelen, there's a huge polymorphism with really split populations with very different body dimensions and body masses and ecologies. And I was wondering whether at the size of Kerguelen you could see any trends or differences between at the subpopulation level.
So not necessarily between, so both of our sites from Kerguelen were provided in 2 different sets of studies, but they both shared very similar haplotypes within that. So we felt like we had a good representation of that population. I'm not sure if that's your question, because everything that between the 2 colonies on Kerguelen was quite closely shared. But I think if we had finer scale sampling, for example, then you might see more of a signal. Though from my previous work on looking at the Falkland Islands and the population there's quite a lot of admixture, even in such a large population as the Falklands, which is where a large proportion of the Gentoo global population is located. So even though they are phylopatric, there's still a lot of admixture among proximal colonies. So I would expect the same to occur in Kerguelen. But what we could definitely use a lot more of is more accurate census counts from the Indian Ocean. Sometimes it's very difficult to, I found a lot of difficulty in getting good, accurate counts for the global population of Gentoos. I know that that's something that even, it might seem very basic, but would definitely help a lot across seabird scientists, is if we had better population census counts from these various colonies and various species.
Yeah, but I think the Kerguelen story shows, how little, well, for Gentoo penguins, how little dispersal there can be even at smaller geographical scales. So that's probably an interesting case. And otherwise, it was more a general comment. I was really fascinated by the side of the story which dealt with the evolutionary history of the Gentoo penguins, and each time, I'm struck by for how long they've been around, millions of years, and also, how they colonize the different areas, and I think, always remembering for how long they've been around, that helps put lots of the debate about, for instance, climate change impacts or global change impacts at smaller time scale into perspective. But that's more of a comment. So that's really the part of the study I enjoyed most, I must say.
Yeah. Cool.
Thank you.
Thanks, David. Thanks, Hila. If we can get you to stop sharing your screen there, perfect. All right, I think we've got one question from Abdullah again. Let me unmute you, my friends. And your hand is down. Go for it. Wait. Hang on, it's taking a second. Oh. It's a little bit slow on the uptake sometimes. Come on, button.
Can you hear me?
I got you now. Okay.
Right. Thank you very much for Hila for the paper. It's opened my mind to a new thing about immunology, which is I'm not very good at. But just to, I was browsing for another paper on African penguins published in 2016 in PLOS ONE, and that paper was dealing with the same TLRs, and they found that there are differences between ex situ and in situ SNP diversity. Do you think if your study in future include also those penguins of your species complex found in zoos in North America or in Europe, you might find similar trend where those individuals at zoos have a lower lower SNP diversity, single nucleotide polymorphism diversity?
I think that's a good question. Okay, sorry.
And the other thing very fast is species complex in your abstract. I've just seen the abstract. Is that means that in your population of study, you have more than one subspecies?
Yes. So first, I think what I can do to address the second question is to share this this like little graphic that I made to show how there has been a debate over taxonomy in Gentoos for the last 100 years. So so right now, what we're mostly operating under is this 1970 construct of 2 subspecies, where everything north of the polar front, including the Indian Ocean, is Pagacellus papua papua, and everything south of it is the southern Gentoo, Pagacellus papua ellsworthy. But a paper that myself and Gemma Klukas wrote in 2016, a paper that Juliana Viana and Deli Knoll and her team wrote in 2017, and then Gemma Klukas's lead paper using genomic SNPs in 2018 have shown, again, there is just greater support for these Indian Ocean clades to be separate and to designate more subspecies. And I know that there's a forthcoming paper, hopefully this month, in Diversity and Distribution, which also uses genomic data in penguins. And I know some of those authors are on here. That's also led by Juliana Viana and her team. So that's, again, more support for separate clades. And that finally also did genetic sampling of Macquarie Island, which is off of the coast of Australia. So that's the first one to really get data from all of the different sites and components of Gentoo penguins. So I do hope that in the next few years we see a greater expansion of how we consider the subspecies from a genetic standpoint, and not just from a dimensions or appearance, you know, how we used to measure their beaks and wings, but look at what the actual diversity is. And then to answer your question about zoos and about the African penguin paper. So I'm also happy to mention that Stephen Fiddeman, my co-first author, who looks specifically at comparative immunology, is working on a paper on all TLRs, so all 10 TLRs in birds and across all penguin species. And that comes from the recent penguin genome papers that were published in the last 6 months or so. And so using that data set, we have some interesting results of the differences in diversity, the differences in these genes in penguins as a whole. But that's not all population data, that's just, you know, single genomes. But with population data, we did find some interesting trends in crested penguins for one particular type of toll-like receptor. So hopefully that'll be coming out in the next, you know, year or so, hopefully soon, but yeah, I'll give it a year. So for zoos, I think a lot of the populations that are currently in zoos have probably been brought into captivity. I don't know, maybe 50 to 100 years ago. They're not necessarily very recent. And so I would actually expect to see some probably highlighted diversity, because a lot of the penguin species that are in zoos are exposed to many more pathogens and different pathogens than what we see in the wild. We know that avian malaria, for example, wipes out penguins in captive settings frequently. And there's many case reports from the veterinary journals that show the kinds of diseases that captive penguins are exposed to, like aspergillosis, which is fungal, and can activate TLR15 potentially, malaria, other hematoprotozoans. So I think that it would be interesting to look at what the diversity is like in the current world captive population. That's a good question and a good research direction that maybe somebody could take up.
So you expect a higher diversity in zoos because they're exposed to more pathogens than in the wild?
I mean, they don't have the same evolutionary timescale with which those pressures and signals would probably be detected. But I think that the pressure is stronger, is what I would expect, because they're probably less adapted. And so we would see a strong pressure. It's just, have we had enough time for them to show that in their offspring? And because it's such a limited gene pool, I'm not sure that we would get that result either.
Yeah, the thing is when you have a lower haplotype diversity in zoos, sometimes that one, and of course the shorter evolutionary time, that will not maybe be representing the real situation.
Yeah.
Thank you.
Thank you. That was great. Do we have any more questions from the audience? I don't see anybody putting their hands up at the moment. Levy, do you have any more comments or questions for Hila?
Yeah, that's really a question from a non-specialist. But so I wanted to get back to the differences between the approaches you take here and I would say more conventional genomics, for instance. How would you explain the differences to a layperson about the two approaches in terms of working out the phylogenetic history of the species, for instance?
Sure. So we still stuck with a very conventional approach for figuring out the phylogeny of the species, and that's because we needed something to ground our work and our comparison with. And so mitochondrial DNA are maternally inherited in most vertebrates. And so what we were able to show is it's this rapidly mutating marker. So it's just one very small part of the genome, and we can look at that, but we have something, because it's so studied, we can measure it and compare it across many other species and populations. And so we've chosen that as a common marker. It's also commonly sequenced. It's easily sequenced. And so it's short enough that we could represent that and share it across research groups very easily to build our tree. So we've done that, and I would say that is quite conventional versus doing a whole genome approach where it would be a lot more difficult to align. We've had difficulty even in just some of the TLR genes, just trying to align them, because across species, when you move into greater evolutionary timescales and differences, some genes can be multiplied or duplicated, and some genes stop being expressed. They become pseudogenized, and that's what we're finding a little bit in crested penguins with one of the TLRs. So in birds especially, it's quite well studied how these genes can change. They can appear out of nowhere, they can be duplicated, and they can be reduced in size. So we didn't necessarily try to build these trees because we don't know the mutation rate. We don't know the time scale at which these particular genes and markers could change over time. So all we can say is this is the current snapshot in time, and this is the current diversity of these immune genes right now. But we can differentiate them, and we can see how many steps or differences were likely to have occurred between them. And we can just measure that diversity, and then we can say, okay, it's clear that here there is less diversity. Is that lesser diversity the same as what we see in other parts of the genome? And in some cases, it didn't match, right? Because one possible explanation for why is there so little diversity in the Antarctic for our particular genes might be, well, they were hopping down the peninsula over time, and so it's just a population bottleneck or a founding population. But that didn't match up with what we knew from other parts of the genome, which were being maintained at a certain rate and with a certain diversity. So this was very clearly just this one gene is having some kind of pressure on it evolutionarily. So we do have to use a combined approach, because you can't, the why or the how is very difficult to determine in genetics, because there's statistical ways that things can come about through chance or through selection pressure. So we did have to do that kind of comparative analysis to make sure that this was not the other, what is the null hypothesis? So trying to figure out that this was actually some kind of other differentiation that occurred.
And maybe a last one. So you put forward the sentinel aspect in the abstract, and I was trying to follow the logic, because in this case, basically, that the birds have been challenged by something, right? But you don't know necessarily by what. So in terms of using them as sentinels, I was trying to figure how it would work, because you would detect that they react to something, but what? So from that point in time and in terms of management, you would still have to be looking for the pathogen to then take measures to reduce that source. Is that right?
Yeah, so from a sentinel aspect, it's because the species as a whole, not just in the context of immune genetics, is a sentinel species of what's going on with our oceans, as you all know, for all seabirds. And so I think what we are advocating for is differences potentially in management, because there are different pressures. And so for us to consider the species as a whole as not differentiated and having the same pressures worldwide is clearly not the case. Whether that was in a distant past or a recent past, there's some difference, some debate in what that might be. But what's very clear, and I think we can all agree across humanity now, is that there is a need to look deeper into the diversity of pathogens that are present in wildlife. And we just don't know enough about what that is, and what is normal versus introduced, what is alien to this environment, what may have been brought by humans, what may be passed back to humans, and what may be shared across flying seabirds and migratory seabirds. Because we know, for example, seabird ticks, right? They're brought across hemispheres. They're brought down into Antarctica. There's arthropod-borne viruses in ticks that are being detected, in recent studies. So there's clearly a lot more global interaction, even in these remote seabird colonies, with pathogens than I think people might realize. And so there's certainly a need for greater research in that regard.
Okay, thank you.
Great. I don't see any more hands up from the audience, so it's 6 o'clock here now in the UK, 5 o'clock GMT, and with that, I think we'll thank you very much for joining us, Hila, and congratulations on a really cool paper and some really good work. I love Gentoos. I think they're totally awesome birds, super plastic, super awesome divers, swimmers, love them. And thank you, Camille, for joining us. And again, thank you, David, for co-hosting this Seabird session with us. We'll be back again next week to our Australia, New Zealand time zone, which will be 6:30 in the morning GMT or 7:30 here in the UK. And please join us again for that if you can. Thanks again, everyone. Nice to see you all. Stay safe, stay inside, wash your hands, don't get sick, and take care.
Thank you, Grant and David, for organizing, and thank you, Camille, for your presentation as well.